Good afternoon, everyone, and welcome to what I believe is genuinely an exciting milestone for the APPS community. The very first APPS seminar hosted here in Canberra. My name is Zakil, and I have the honor of serving as the ACT Regional Counselor for the Australian Plant Pathology Society. This seminar series is something I think we can all be proud of. Starting today, each state and territory will take turns hosting a monthly event every second Tuesday of a month, bringing together plant pathologists and allied scientists from across the country and the Pacific region.
Hello?
Hello, this is
Am I
speaking.
To share research findings and ideas,
Just
spark
hang on.
collaborations.
I am in another meeting, so I just gotta.
Somebody is speaking in the background. To share research findings and ideas, spark collaborations, and celebrate the excellent work happening in the field. The ACT is proud to kick things off. Today we have two outstanding speakers whose work spans the breadth of modern plant pathology, from harnessing the power of soil microbes to deploying artificial intelligence in the field. Our first speaker for today, Dr. Louise Thatcher, is a microbiologist and molecular plant pathologist based right here in Canberra at CSIRO Ag and Food. Louise leads research on biopesticides for crop protection, and her work on beneficial microbes and their natural products is addressing some of the most pressing challenges in sustainable agriculture, from resistance development in conventional crop protection to emerging biosecurity threats. Today, she will take us through her team's work on Australian Actinobacteria and how multi-omics approaches are being used to unlock their potential for crop protection. Our second speaker for today is Dr. Manish Patel, a research scientist with CSIRO Ag and Food, based in Canberra. His research focuses on cotton disease phenotyping using computer vision and deep learning. He completed a joint PhD at the University of Melbourne and the University of Sydney, specializing in multispectral and hyperspectral remote sensing for crop nitrogen sensing. His broader interests include object detection, segmentation, drone sensing, precision agriculture, and remote sensing. Manish will take us into the world of computer vision and deep learning, and he will share how he has developed a field deployable imaging pipeline to identify and quantify verticillium wilt in cotton stems, a solution that eliminates operator bias, dramatically increasing throughput for breeders and opens the door to drone-based disease monitoring at scale. Each speaker will present for 15 minutes, followed by 10 minutes of questions. So please do have your questions ready. Let's make the most of the time we have with both of them. Without further ado, please join me in welcoming Dr. Louise Thatcher. Louise?
Thank you, Zakil, and thank you, APPS, for this opportunity. So I'm going to give you an overview today of some of the work that we do in the microbial technologies team. Let's start out with the threat. So this was a bit of a snippet from the internet that the real fungus threat isn't a zombie apocalypse, but really it's fungal pathogens attacking crops and agriculture. And we know that fungicide use and pesticide use in general has been increasing over time, yet crop losses continue. And we see the development of resistance in pathogen, pest and weed populations. Now, we're not immune to that. In Australia, we've seen multiple fungicide resistance appear across the country, and it's not limited to fungi. We also see the same in insect pests, such as the green peach aphid here. I think if someone could just mute in the background. Thanks. So we have these resistance challenges, but we also face what we call a chemically constrained future. And so we're losing effective chemistries, but we're also seeing a slowing down in new chemical modes of action that are coming to market. And linked with this, we see some traditional crop protection products being removed from the shelf, but there's also increasing calls for sustainable farming systems and global market drivers that we need to face. So biopesticides and biological agents are one option that we can use in crop protection with all our other integrated disease management options. So these include, on the left there, you can see in this little picture, they may be beneficial plant-associated microbes. They could be microbial pathogens of pests and weeds, and they can also be the natural products derived from these living organisms or plants, as well as beneficial insects and natural enemies. But there are challenges in adoption of these solutions in Australia. One is we need more novel products for Australian growers. We have limited number and diversity of biopesticides registered in Australia. And obviously we're also restricted by importing living organisms because of our strict quarantine. We also need sovereign bioproduction at scale with formulation. We know that decades of research has gone into formulating traditional chemistries, and we need the same efforts and new technologies into formulating biological options. And another critical challenge is how we integrate these products into farming systems and making sure that we have field protocols and performance indicators that are relevant to the biological modes of action. And they're not field protocols and performance indicators just lifted from what's been developed for traditional chemistries. So at CSIRO, I'm a part of a larger group of researchers across the country working on these biopesticides. And we aim to develop new biological products to manage pests, weeds, and diseases, along with the tools and practices that are needed by growers and advisors that work at scale in our current and future farming systems. So today I'm going to go through part of the pipeline of work that's done across the organisation from discovery through to deployment. But I'm going to focus on just the discovery and improvement side and give you some examples that we're doing in the team. And it's all based on Actinobacteria. So why Actinobacteria? Well, we chose these very interesting microbes because they can form beneficial interactions with plants, but we can also extract their bioactive compounds or molecules. And they have very high biochemical potential, known to produce several thousand structurally diverse molecules. For example, as shown on the schematic on the right, they can produce fungicides, insecticides, compounds that activate or prime plant defense responses, as well as producing molecules that interfere with pathogen communication or colonization onto a host plant. And because they exhibit this broad range of activities and different modes of action, we typically think that they would have lower propensity for the development of resistant pest populations. But there's still several unknowns. We know they can produce these sorts of molecules, but we know very little about what these individual molecules are. If we look at bacterial genomes that are publicly available, the Actinobacteria represent a very small proportion of these, which means we can't really look at what the diversity that's available. We know from the genomes that are sequenced for the Actinobacteria, they're predicted that about 90% of their biochemical potential is yet to be discovered. So this is really exciting for us to look in here for new molecules and new modes of action to protect crops in our agricultural systems. And commercialization pathways already exist for Actinobacteria, so we don't have to reinvent the wheel. We know that we can produce commercial products. And there are examples, I've just shown a few on the screen at the moment that represent biofungicides, bioinsecticides, or bioherbicides. And they may contain the living actinobacteria, a filtered extract, a purified product, or maybe now they've been produced synthetically. So how do we start developing a collection of actinobacteria to screen? It all started with Dr. Margaret Roper, shown in the bottom left of your screen, with help from GRDC. And what Margaret did is developed a very targeted collection of actinobacteria that was suitable for agriculture. So firstly, on the left, she used very targeted locations. So she selected areas in agricultural production areas, and so we identify locally adapted strains. So this is very important that the strains can grow in that region, and we're also not affected by those quarantine issues if we want to import an organism. She also conducted targeted sampling. So she talked to growers, and she identified areas of paddocks that were always well performing, had healthy plants and non-disease crops. And then lastly, she did targeted isolation. So she took wheat roots from those paddocks and she isolated the actinobacteria endophytes within them. So from this, we would assume that those actinobacteria are already forming very positive relationships with those plants. And now I'll go through some of the discovery and improvement examples that we've been doing in the lab. The first part, in terms of discovery, we've been screening those strains or looking at consortia, so multiple microbial strains, as well as identifying their natural products or bioactives that give them their activity. We're very much interested in those that can provide new modes of action. So that's to control any pesticide or fungicide resistant pathogen populations. We're looking for strains or molecules with improved efficacy, potency, stability, or durability. So an example here, one of the streptomyces that we identified from the collection is a highly effective biofungicide. Shown here is some seed coating where we've treated these wheat seeds with our streptomyces and then challenged with Fusarium pseudograminearum or crown rot disease of wheat. You can see here in this panel D that the plants are free from disease and comparable to the non-disease controls compared to these diseased plants or ineffective streptomyces inoculant. When we take that same streptomyces, in this example, we've looked very early during challenge with this case Rhizoctonia solani fungal pathogen. You can see this zone of inhibition very early on after being challenged with the fungus. And so we can't even see the streptomyces on this seed. So this tells us that the molecules that it's producing are very potent and able to protect that seed from the Rhizoctonia. That same streptomyces we also grew in a shaker flask or bioreactor and developed an extract or filtrate. So we removed the streptomyces after several days of growth, and we only have in that filtrate compounds or actives that have been secreted by the streptomyces into the broth. In the panel on the left, when we sprayed that filtrate onto canola plants and challenged with sclerotinia, we saw very good protection in this bottom part of the panel, and indeed it was comparable to the synthetic fungicide Prosaro. In this case, we sprayed the plants at 10 and or 30% flowering stage to mimic what growers do in the field. Interestingly, we found when we sprayed with the microbial culture of the streptomyces, we also got good control, but not to the same level as the filtrate. So the filtrate was very active. We saw the same effect when we treated a different plant against a different disease. In this case, Fusarium graminearum head blight. You can see in the left part of that panel, these wheat heads are severely diseased with the fusarium compared to the wheat heads that receive the biofungicide filtrate, virtually free of disease. So then we're very interested to understand, okay, what might those bioactive molecules be in that ferment? And so we used a multi-omics workflow. So we'll go through this. Essentially, we repeated the shaker flask experiment, and we did a time course over several days. Each day we subsampled from those flasks, and we tested for antifungal activity. And this is the sort of result that we got on the left. So what we do is we challenge a fungal pathogen that's been transformed with the green fluorescent protein. So where you see green here means the fungus is growing. Where you see black cells is we've either killed the fungal spores or we've severely inhibited its growth. And so when we took extracts from this time course and challenged it against the fungus, you can see between this 72 hours and 96 hours, we got a very strong degree of inhibition. So that tells us there must have been some molecules being produced during this shift in time that was causing this antifungal activity. So what we did is we took cells, the Streptomyces cells from those two time points, and we conducted RNA sequencing to look at the whole transcriptome. But at the same time, we also did metabolomics analysis to look at what the molecules in that broth are. And this is the results we got back from the transcriptomics. Essentially, we were able to identify regions of the Streptomyces chromosome that were activated during that transition to when it was started to become active in producing antifungal activity. So shown in these little circles is essentially the relative expression of genes in a biosynthetic gene cluster. So you can see that some were highly expressed at this later time point. And if we plot that on the lower graph, again, if we go across the Streptomyces chromosome, here we're looking at if it's upregulated or downregulated genes in a biosynthetic gene cluster. And you can start to see clustering of genes, particularly at the ends of the chromosome that are active in those colors. When we did metabolomics on the samples, we identified several compounds that were highly produced at the later time point, and these encoded two known antifungals, the nigericins and nifamycins, as well as iliophyllin, which is a potentiator of antifungal activity. So that was really exciting, as well as a siderophore compound, which are known to sequester nutrients. So that work, we've been able to use that same sort of workflow to identify new types of molecules in a rapid way. The other work we do in the lab is, okay, we've identified that promising Streptomyces strain. How do we make it produce more of the molecule of interest or grow more fast or grow on cheaper feed materials? Obviously, we need a final product that is relatively cheap to go into an agricultural system. So we conduct strain or molecule improvement, as well as looking at novel bioproduction systems. And just an example here that we've been doing working with New Farm was looking at iterative scalable process development of an actinobacteria. So it all starts in small bioreactors that we can run in parallel to test various feedstocks, time, temperature, etc., to get the microbe to grow more quickly. We then run it through our antifungal assay. Essentially, then we can identify the time points or conditions that give us the most activity. We do metabolomics to look at what active components are in those ferments. We obviously test that on plants. In this case, we're testing against Sclerotinia on Brassica to make sure the in vitro result matches what we want on plants. And then we go through this process of iteration to get more and more bioactivity more quickly and more cheaply. We then transitioned this to larger scale fermenters to see that we could scale that process into something that's relevant to agriculture. And so now we've been using that same pipeline towards other screening for other sorts of traits that would be beneficial for agriculture. For example, screening against insects, looking for phytotoxicity if we're looking for bioherbicides, but also beneficial properties like looking for increased root growth or other plant health aspects. We couple that with, we've sequenced the genomes of all the strains in our collection with complete or near complete genome assemblies, as well as matching out with metabolomics data. And then others in the team are looking at the strain improvement, which could be domesticating wild type strains through non-GM ways or using genetic engineering to improve strains. So I've given you a bit of a snapshot of what we do in the team. A lot of different people are involved in those various experiments that I showed you. Obviously, the different skills and capabilities are needed to work across those different science domains. So big shout out to New Farm, GRDC, the MOSH and Advanced Engineering Biology Future Science platforms at CSIRO, as well as the many state departments of agriculture that have helped us along the way. Thank you, and happy to take any questions.
Thank you, Louise. So we can have two to three questions.
Hello, I am Wadia from Lincoln University.
Hello.
Hello. Yeah, very nice talk.
Thank you.
I just would like to know how far are you concentrating on one single bioactive compound or looking at multiple compounds?
Looking at multiple compounds, yeah, typically what we see in the ferments, like the example I showed you on the screen, typically we find several actives in the ferments. Now, whether we want to narrow that down to producing just one compound or keep it as a mixture, because obviously then hopefully those molecules might work in different ways, and it again help to prevent resistance development in pathogen populations.
All right. Okay. How far are you, do you think, to go into actual farm work, or are you already into it?
Yeah, we already do field trials. At CSIRO, we don't commercialize products, but we hand information over to someone else to produce.
All right. Okay. Yeah. Thank you.
Thank you.
Any more questions?
Yeah.
I have one. Oh, you go.
Hello, my name is Jia Hawk. I work in WA. I work on SBDI project, especially on Sclerotinia and Rhizoctonia aspects. So I am highly interested to using the bioagent for controlling those pathogens. So is there any field usable product or bioconcertia that available to test against Sclerotinia and Rhizoctonia aspects? And I saw some literature that is a Conidia minitans is widely used in US and Canada, but I'm keen to use this product, but it's unfortunate it's not available in Australia. So
yes, so that is one of the challenges that we highlighted at the beginning. So we've been involved in a couple of discussions with the APVMA as well as others, and how we can help streamline the process in terms of registering new products in Australia. Certainly that product does parasitize the sclerote, so it does give you that ability to control within and between seasons, which would be great. Yeah, I think that's a part of the challenge at the moment is getting products registered for evaluation.
that'd be good. So Can we use your product in our research aspects, like the
I guess it's
bacteria?
restricted by IP at the moment, but we could talk offline about that. Yeah.
That'd be great. So we are trying to use some bioagent other than fungicide in our project. So our project is started in 2025 to last year to 2030. So we are highly interested to use some bioagents in our research activities.
Thank you, Zia. There are clearly more great questions here, but in the interest of the time, perhaps we can continue the conversations maybe in the chat box. For now, please join me in thanking Louise, and let's welcome our second speaker, Dr. Manish Patel.
Thanks, Zakil, and thank you, APPS, for this opportunity. And very interesting talk, Louise. Thank you for that. Okay, I'll start sharing my screen. Can you see the screen?
Yes.
Okay. Yeah, so I'll be talking about the computer vision and deep learning approach we are using for disease phenotyping in cotton in CSRO. So it's a joint project between the CST and CSRO. So yeah, my name is Manish. I'm a research scientist in Ag and Food, CSRO. Yeah. So before I start talking about the computer vision and deep learning, let's have a look at like what I mean by disease phenotyping in cotton. So here you can see that there are two main manifestations of disease we see in the cotton. One, on the left-hand side, you see the stem-based manifestation. On the left-hand side is a disease stem, and on the right-hand side is the healthy stem. So the amount of discoloration or the discoloration in the stem after the harvest, it gives us indication of the disease in the cotton stems. And on the right hand side, we have another sort of manifestation of disease, which is like foliar symptoms. So you can see like the leaf curling or like discoloration, or even sometimes the defoliation in the leaf. That can be one of the ways of assessing the disease in cotton. So what's the traditional way at the moment? The traditional way at the moment, because the cotton breeders, they definitely want to select the resistant varieties. And these resistant varieties, they have to first do a field assessment. So these field assessment are usually done manually at the moment. And the problem with the manual stuff is, I mean, not all the manual stuff, but the scoring stuff in the field for stem and foliar-based symptoms, it's very tedious job. It has very limited information. So when I say it's limited information, they only have the information like, like it's a disease or not diseased, and also like the foliar symptoms, it's very subjective as well. So that there comes the operator bias as well. So different operators or different scorer has their own interpretation or their own interpretation of the disease symptom and their severity as well. So what we're trying to do here is like we develop the computer vision AI-based solution to basically do the disease phenotyping in cotton, post, stem, and foliar levels. And why we want to deploy that? Because we get greater scales. So and the speed is higher than the manual-based scoring in the field, and accuracy is higher sometimes, like especially for the challenging field conditions, and it has some positive impact for the unbiasedness. But we do know like the AI models, they have their own biases. But yeah, but it's free from the operator bias. And then the depth information, you can revisit the images later on, or do the scoring again and retrain the model, and you can deploy the same AI model across multiple sites at the same time. So it's also cost effective. So I'll talk about the stem-based disease detection. And in this case, I'll be using the verticillium symptoms in cotton. So I'll start talking about like how we did the image collection and the annotation and then the modeling approach. So when we go to the image collection, that's the first thing, like the model has to learn from the images we collect from the field. And the requirements are, I mean, very simple, but very high, like high resolution or the highest possible resolution, the highest possible image quality, and at high scale. But the challenges we face in the field is like when we collect the images, it's motion blur. Things move very fast behind the stamp cutter or the tractor. And we have a lot of high and low frequency vibrations. We also tackle with dust and small object detection. So on the left-hand side, this is what I mean by the high quality images. So this is the level of images we want from the field, and that's what we get. So this is a GoPro camera mounted behind the tractor, and you can see like after the stems are cut, it's fully action-packed, and everything moves so fast. And you have to have specific equipments to do the imaging in the real time, because if you leave the stems open after cut for a long time in the air, they get oxidated, and then their color changes as well. The next step is, once we collect the images, we have to teach the models like what we want the model to learn and what we want the model to predict for us. So this is one of the steps like we use for annotations. So here you can see like we use thousands of images, or maybe more than thousands of stamps for creating the annotations, and these annotations will be used by the model to learn the features we are interested in. So this is one of the examples like how we do it in manual mode. And yeah, it takes a lot of efforts and time and resources for doing and generating those sort of data sets. And once we have the images and the annotations are generated or created, then we train the models. So models are basically trained like a toddler. So like the toddler, how it learns to step up and the walk. So it fails a lot of time, and then sometimes it. Can you see the animation? No?
We can see something.
But, can you see the animation?
Not the animation.
Not the animation. Right. Yeah, no. So you can see like this is one of the field images we collect from the field using the handheld cameras. So it's very accurate images, and you can see like how the model starts learning from it. So if you see it from here, earlier it starts detecting everything. Again, the animation is gone. Sorry about the animation. So what I was trying to say here is, so you can see animation now. So the model starts with a lot of errors at the beginning, and you can see like the model will basically detect everything at the start, like even the cotton balls and the debris. But as the time passes, or as we have more iterations, the models start converging towards the lowest error, and that's where the model learns better. So we do give or feed in a lot of information in terms of images, annotations, so that the model learns the more generalizable way of detecting the stamps, and as well as classifying them into disease versus healthy stamps. And in this video, I'll show you like this is one of the data set we recently collected from the field right behind the stamp cutter or the tractor. you can see like we run the model for the whole range of images as a video, and you can see like the model is not detecting any debris at all, and it's scanning through all the images, and it will start detecting once it sees the actual stem, and then it classifies them into disease and healthy ones. So, and this is like 1/30th of the speed of the real-time speed of the camera system. And you can see like the model is still not detecting anything because there is no stem. It's all debris around. And you'll see now you can see like the model has started detecting the stems. So even we can't see some of the stems in from our eyes, the model is able to detect them and classify them into disease versus healthy. So that's what we wanted, and we are still improving the system. It's not the perfect system at the moment. Still misses some of the stems and still misclassifies some of the stems into disease or intermixed them. And we have published this results this year in a journal. So yeah, feel free to have a look by using the QR code, and then know more about like how we develop these models and what are the images, how the data set look like, and what are the steps involved in developing this sort of models for disease detection in the field. So to wrap up everything for stem-based disease detection, we have this tractor, and we have a stem cutter mounted behind the stem cutter. We have a lighting system and the camera. We use them to detect or collect the images, and these images are fed to the trained model, and the trained model detects the stems in the field images and then classifies them into healthy and diseased ones. And these information are then fed to the then share with the breeders, and they use this or they're going to use this for the resistant variety selection. So this far we have come for stem-based disease detection. The next few slides I'll show you about the initial steps or the data collection we are doing for foliar-based symptoms. So we are basically looking at the canopy level symptoms, and for that we're also using the computer vision AI-based solutions. So one of the ways we're using is remote sensing and the drone-based or drone-mounted cameras. So here you can see it's one of the videos from the field, cotton field, and I was flying a hyperspectral drone there. And on the bottom left, you can see one of the cotton experimental field site with a lot of cotton trials. And once we collected the images, so this is one of the cameras we use. We call it multispectral camera. And that camera gives you 5 channel information. So blue, green, red. Blue, green, red, we can see from our normal cameras. Even our phone camera have the blue, green, red. And then the red edge and NIR. So red edge and near infrared, which is NIR, is the extra bit of information we get from the multispectral cameras. And here VW means verticillium wilt at the canopy scale. So this is the VW or the verticillium wilt score given by the trained or experienced breeders in the field. And we were plotting the reflectance, which is collected by the drone-mounted camera, multispectral camera. You can see like there is an apparent difference between the different verticillium wilt scores across, and this data is across different growth stages. So there will be some overlap as well. So, but there is apparent difference between the reflectance values of different levels of VW scores. And in the previous slide, I was talking about the multispectral camera where we had 5 channel information: red, green, blue, red edge, and infrared. And in this one, we have a hyperspectral camera. So we are collecting the data from the hyperspectral camera we call Molnier. It has a spectral range from 400 to 2500 nanometer. So basically, like in the last slide, I talked about like 5 channel information, and then in this one we have 300 channels. So that's the difference between the hyperspectral and multispectral. On the left-hand side, if I give you a picture, graphical representation of hyperspec, you can see like the spatial dimension we have X and Y, but the spectral dimension, if you go with the same field, like this is a current field, the spectral dimension we have up to 300 to 400 channels. So it's a 3D image, or the 3D information we collect from the hyperspectral images. And why we go for the hyperspec? Like I'll give you one quick examples. Like it's if you have a water feature, if you have like some earth surface features in the field, soil, water, and vegetation, they have their unique spectral signatures. So if you go with the vegetation or the crop, they have this typical spectral reflectance. And if you have water, it has a different spectral signatures, and the soil different as well. So these spectral signatures, they change with the property or the current state of the feature as well. And the change is what we are interested in to capture using the spectral information or the hyperspectral camera. That will reveal a lot of insights about the Earth surface features we are interested in. And in our case, we are interested in vegetation only. So here is another recap. So it's just like a brief information. We are still processing the multispectral and hyperspectral data. Probably in some time later, I'll have a more interesting information or the stories to tell you about the multispec and hyperspec. But so far, just a brief introduction. So when I say RGBs, this is the standard color cameras we have, DSLR or mobile phone cameras. We have only 3 bands or 3 channels or 3 information, let's say. When we go to the multispectral, in our case, we have 5 channels. So we have 5 distinct bands. But when we go to the hyperspec, we have basically hundreds of channels. So in our case, we have 300 to 400 channels, depending on the binning we're doing. And that gives you a lot of information about the insights of the vegetation. And I'll give you one quick example. So this is one of the cotton fields we have in Malwell. And there are 2 populations we have. So 22SF1 and 2269. So on the left-hand side, you see the normal RGB image from the drone. And you can see like it's a bit difficult to, differentiate between the 2 populations. So, but when you use the multispectral models, it's much easier to differentiate between the 2 populations. So 22SF1 and 2269. So you can see like the 22SF1 is more resistant towards the verticillium wilt in the field. So that's what we are interested in. And also this will give you the quantitative information as well compared to the qualitative or like only like 0 to 5 scale one. So yeah, that's pretty much from my side. And yeah, it's a team effort. So a lot of people are involved. And yeah, I'm being the face of the presentation, but yeah, it's a team effort. Thank you very much.
Thank you, Manish. Please join me in thanking Manish. And questions to Manish. Manish, I have a question.
Yeah.
From your first part of your talk, do you think simple detection of disease versus healthy stem is sufficient for the breeders, or would they need something like quantifying disease severity in a sort of 0 to 100 range or whatever?
Yeah, I didn't show the slide, but yeah, we do have a work going on for quantifying the disease after it's detected, like disease versus healthy, because disease versus healthy is still a limited information. It's still like just a binary one, but how much diseased, even if it's diseased, that will give you more insights into the resistant, the disease resistivity of the varieties. So yeah, we do have a work on it.
There's a question in the chat, Zakil.
Yes, I can see. So there's a question: how many images were used for the annotation, and when the machine has finally phenotyped the disease and severity accurately?
So in the initial phase, until last year, we used somewhere around 5,000 images, and 5,000 images have multiple stamps in it. So I think, so the paper I shared, it has all the information, but I think it should be around, more than 5,000 stamps as well, somewhere around. So we use that information to train the model. And once the model was trained, we tested the model across different sites and different year of the collection. And we found that the model performance on the independent data set was somewhere around 80 to 85% mean average precision. We call it in object detection world. So it's pretty promising result. Like if you compare the contrasting or the challenging field condition we have in the field to get the images and do the detection and also do the classification. So yeah, it's pretty promising in terms of accuracies and the results we are getting with the model.
There's one more question from Kit Jaisena. The system you use, can it implement for threshold-based fungicide application?
What threshold is it? Image based? Depends on the symptom. If the symptom is it's like illumination dependent or the camera dependent, or like if the symptom is varying for different time of the day, I don't know like which type of symptoms, but then you have to plan accordingly and you have to train the model to circumvent those changing situations.
More questions coming.
Mm-hmm.
What value do the growers find from post-harvest disease detection? Also, once plants show foliar symptoms of verticillium wilt, do growers have management option to reduce the impact of disease?
That's a good question. So for the growers, like post-harvest disease detection won't give them much value, but for the breeders, this give them a lot of insights to select or get the more resistant varieties that ultimately helps the growers at the end. So it's a cycle that will definitely come back to the growers at the end after this work. And regarding the before harvest any disease management, yeah. So regarding the foliar symptoms I was showing, you can use that foliar symptoms to check the disease patches in your field during the growth, and then you can use any mitigation steps or any ameliorative steps or like the fungicides to control that.
Thank you.
I'm not aware of like, because it's very disease dependent. Like for verticillium, fusarium in cotton, I'm not. There may be some chemical options that you can apply, but I'm not expert in that field. So yeah.
Manish, could you please put the final slide?
Yeah.
So while thanking both of our speakers today, the next seminar will be on 14th of April. That will be hosted by the Northern Territory. And so that the more information will be available at the APPS website. Having said that, I want to tell you that if you are not yet a member of APPS, we warmly encourage you all to join the society and become part of our plant pathology community. And thanks for joining us today.